examples. So in brain imaging studies, people image brains under, say, null conditions, controlled conditions, and then under some kind of stimulation conditions. And they're interested in which parts of the brain are responding to a stimulus. So you can think of the data as being images. They're actually often 3D images because the scans are 3D. So if it's 2D, it's pixels. In 3D, they call them voxels. But if you're not used to 3D, just think about an image, 2D, a 2D image. Each pixel, you're testing the null is that pixel, the part of the brain in that pixel is not responding to the image. And you want to test that null for every pixel in the image. So that's an example, where you've got hundreds or thousands of pixels, or usually thousands of pixels. And so there are thousands of tests. In genetics, where I work, we're often testing different parts of the genome, or often different genes, to see whether they're different between, well, again, sometimes it's between cells that are in a control condition versus cells that have been stimulated. You stimulate the cells with something, you want to know which genes are responding to that stimulus as a way of understanding the system. So for each gene, you have a null hypothesis that there's no response. And an alternative hypothesis is that it responds. And you want to test that for every gene. And there are roughly 20,000 genes in the human genome, depending on how you count. So there are 20,000 tests. And I don't know, people often talk about the burden of multiple testing and the challenges of multiple testing, but actually I think of multiple testing as an opportunity, because if you give me just one test, then as I've kind of already outlined, it's kind of difficult to decide what p-value threshold to use, for just one test. But when you give me a whole bunch of them, thousands of them or hundreds of them, then there are kind of, the way I think of it, there's more you can do. One thing you can do is to use empirical Bayes methods. But before talking about those, I want to talk about a kind of frequentist approach to the problem, or at least it's kind of somewhere between maybe empirical Bayes and frequentist approach to the problem, that John Story developed, called Q-value. So essentially, maybe I can just give you an idea of what this approach does just by drawing a picture. And maybe we can, the point here is to try to emphasize the benefits that or the opportunity you have when you've got a lot of tests. So if you give me just one p-value of 0.05, I find it very hard to decide, or one p-value, I find it very hard to decide what threshold to use. But if you give me a lot of p-values, say 100 or 1,000 p-values, I can plot a histogram of them. And what I'm hoping that histogram is going to look like is something like this. Okay. That is that it's going to be enriched for small p-values compared with a uniform distribution, which we might draw like this. Okay, this is my uniform here. So, okay, so one thing you should know, if you don't already, is that under the null hypothesis, p-values are uniform, or possibly enriched towards 1, but no worse than uniform. So your ideal is that a p-value will be uniform under the null hypothesis. So if you've done a lot of tests, you're hoping that some of them are non-null. If they're all null, you expect to see something that looks uniform. You're hoping that some of them are non-null, and so you're hoping to see an enrichment of small p-values that indicate an effect. So you're hoping to see something like this. And when you do see that, you could ask me, well, let's suppose I draw my threshold at 0.05 here. If I do that, what proportion of the tests that I reject are non-null. Right? So what's the proportion of tests that are less than, say, 0.05 in this case, that are non-null? And loosely speaking, it's not quite a precise definition. Loosely speaking, this is referred to as the false discovery rate. We can see a precise definition later, but loosely speaking, the false discovery rate at any threshold is the proportion of tests that we reject. Among the ones we reject, what proportion are actually null? They're the false discoveries. So the naming comes from the idea that when you reject a null, you've made a discovery. You've discovered a gene that's different, or a pixel that's different between the groups. So what thinking of a rejection as a discovery, you're asking, among the ones we rejected, which ones are false discoveries? Which ones were really null? And what's that proportion? So the idea, the way I view it anyway, is that because we've got lots of p-values, this histogram essentially allows us to estimate that quantity, that false discovery rate from the data. And the way that this approach called Q-value does that, so Q-value is an R package by John Story. And the way that it does it is to say, well, you know, this histogram here is a mixture of null tests and non-null tests. And I don't know which ones are which, and I don't know what the non-null tests look like, but I know what the null tests look like. They're going to be uniform. The p-value should be uniform for my null tests. So I can conservatively estimate that, you know, that break this histogram down into a uniform component and a non-uniform component, basically. And that's as high. I mean, it could be that everything's non-null, right? It could be that my alternative is just the same as what I'm seeing here, the distribution under the alternative, that they're all alternative. But if the idea here is I'm going to be conservative by drawing that horizontal line that corresponds to the uniform as high as possible. And essentially by looking at how many p-values there are in this right end and near 1. You know, I can't draw any higher than that up here, because otherwise, if it were there, I would have seen more p-values here, right? Which I didn't. So the histogram kind of limits how high I can draw that uniform. So I'm going to draw it as high as I can to be conservative. That's the idea. And then if I want to give, you know, if I'm going to use this threshold here of 0.05, then the areas here under the histogram in these two parts, let's call this A and let's call this B, then my false discovery rate estimate is going to be, well, the B of the null, the area B represents the null tests. So the false discovery, they're the false discoveries, right? So it's B over A plus B. So the beauty of this procedure is that, apart from being simple, is that it's adaptive to the results of your experiment. So for example, if you actually saw a histogram like this, it's a bit too good to be true maybe, but suppose you did an experiment and saw your results like this, and you drew a line here, and you asked, well, what's my estimated false discovery rate? Well, you can see that what would happen is it would basically assign all the tests to be alternative, and the false discovery rate would be estimated to be about 1, all of them. So the point is, it's that this histogram that's very flat essentially implicitly tells you about things like how strong are the effects that are non-null and how powerful is your test. Those things that you, if you were doing one test, have to make assumptions about, when you're doing a lot of tests, you can kind of essentially use the results of that test to learn about the power of the test and the effect sizes and some combination of those things. And when you see a uniform histogram of p-values here, you know that either you did a very unpowerful test or the effect sizes are very small. It doesn't really matter which, you can't tell the difference. And so this procedure ends up saying, well, you know, whatever your FDR, whatever your threshold, you can't be confident that you've got a low false discovery rate. On the other hand, conversely, if you have a very strong, strong signal in your tests, and most of them are showing a very strong signal, then the false discovery rate, even at a quite, even at a quite liberal threshold of say 0.1, will still be quite small. And that's, that's right. If you do a series of tests and most of them are non-null and they have big effects, then most of the ones with small p-values will end up being non-null. And so just to repeat, it's like the problem with one test is that we need to make assumptions in order to map a p-value of say 0.1 to a meaningful measure of how strong is the evidence. Whereas when you've got lots of p-values, you can essentially use them to make more informed decisions about, well, at any given threshold, what proportion are likely to be true versus false discoveries. So are there any questions about that big picture idea, or indeed any of the other details?